In our lab, we are interested in understanding the mechanisms controlling the development of the cerebral cortex, and in that context, we've been studying over the last few years mechanisms controlling axon guidance and neuronal migration. One of the families of molecules that we've been studying are roboreceptors, in particular Robo1 and Robo2 molecules. And we have assigned some functions for these classical guidance receptors in the context of axon guidance and cell migration. Now, by looking at these double mutants for Robo1 and Robo2, we found a number of years ago that the brain of these animals are smaller. We thought this was very exciting, and naturally Victor decided to go on and study this.
In the developing cerebral cortex, primary progenitors can divide in three different ways. They can divide to multiply and increase the number of progenitors, they can divide to generate postmitotic neurons, or they can divide to generate intermediate progenitor cells that will divide again in the subventricular zone to generate two neurons.
The first we observed in these Robo1 and 2 double mutant embryos was that there was a slight deficit in cell divisions of those primary progenitors, but not in cell divisions of those secondary or intermediate neurogenic progenitors, and also no change in the numbers of neurons. Now, surprisingly, we did observe a twofold increase in the numbers of those intermediate progenitor cells.
With these results, we reasoned that roboreceptors will be involved in controlling somehow the balance between progenitor cells or the dynamics of progenitor cells in the developing cerebral cortex.
To study the dynamics of progenitor cells at the single cell level, we performed a clonal analysis of individual cells in the developing embryo.
With the aid of an ultrasound microscope, we injected GFP-expressing retroviruses in the brain of developing mutant and control embryos to label individual progenitor cells in the ventricular zone of the cerebral cortex.
From these single-cell lineage experiments, we found the same results as we had obtained before at the population level, which was an excess of intermediate progenitor cells, but the same number of neurons. In addition, to our surprise, we also observed an increased number of intermediate progenitor cells with an apical process. Now, at the time, this came much as a surprise because it had not been described in normal embryos. So, the presence of these intermediate progenitor cells with a long apical process, we found it was very abundant in the Robo1 and 2 mutant embryos. And while it was present also in the control litter mates, it was much less abundant. These observations led us to conclude that under normal circumstances, during the formation of intermediate progenitor cells, these go through a transition phase of apical process retention, followed shortly after by a complete retraction, preceding cell division and finally neurogenesis. So, robomutant embryos accumulated too many of these intermediate progenitor cells containing a long apical process. So, an open question that remained unanswered was whether this phenotype was really due to a systemic defect or was cell autonomous. To answer this question, we performed in utero electroporations in control and mutant embryos using gain and loss of function tools.
At this point, we decided to start searching for downstream effectors of the possible function of roboreceptors in the context of neurogenesis in the developing cortex. And for this, we screened through genes that were somehow related to neurogenesis or proliferation in the past, like the Wnt pathway, the Notch pathway, and other effectors.
We found that robomutants had reduced levels of HES1 expression, a classical readout for the Notch pathway. Differences in HES1 appear to be at the core of the increased number of intermediate progenitor cells in robomutants, as revealed by gain or loss of function experiments.
Finally, we tested whether robosignaling might directly influence HES1 transcription. By using luciferase experiments, we could demonstrate that indeed roboreceptors can directly drive the transcription of HES1. So as a conclusion of this part of our study, we could say that Robo and Notch may act synergistically to drive the expression of HES1 in the developing cerebral cortex.
In summary, we found that roboreceptors regulate the balance between progenitor cells in the developing cerebral cortex. We also found that this control over neurogenesis by Robo is mediated through the expression of HES1.
Our experiments identify a new function for roboreceptors, best known as classical guidance receptors involved in axon guidance and in cell migration. Our findings reveal that roboreceptors also play a role in neurogenesis, most likely controlling the balance between different classes of progenitor cells. This function has been previously assigned to other guidance receptors, like for example EPH receptors and efferents.
Yeah, and our findings also have profound implications in brain evolution, because now roboreceptors become strong candidates to participate in the extraordinary expansion in the numbers of intermediate progenitors and neurons generated in development, and that this expansion occurred during mammalian brain evolution all the way to the human lineage. Research in our lab will now certainly focus on further understanding the role of roboreceptors and other molecules in the evolutionary expansion of the mammalian cerebral cortex.